In this video, we'll learn how to describe the structure of the nucleus in a eukaryotic cell. We'll also explore the structure and function of chromatin and chromosomes. So let's dive into the exciting world of the nucleus. If you've ever looked at onion skin cells under a microscope, then you've probably seen something a bit like this. Here we can see all the individual cells that make up the onion skin, and the little dot that we can see inside each cell is the nucleus. But what actually is it? The nucleus is a membrane-bound organelle which contains an organism's genetic material, its DNA. Remember, an organelle is just a subcellular structure that carries out a particular function. The nucleus of each human cell contains a massive 2 meters of DNA that is tightly packed and organized. If we took all the DNA strands from all of your cells and put them together end to end, it would measure up to 6.2 billion kilometers. That's more than 41 times the distance between the Earth and the Sun. Now let's have a closer look at cells. All cells have at least a cell membrane and some cytoplasm. But not all cells have a nucleus, for example bacteria. We can use this information to divide living organisms into 2 groups. Cells that contain a nucleus and other membrane-bound organelles are called eukaryotic cells, and cells without are called prokaryotic cells. Because this video is all about the nucleus, we're going to be concentrating on eukaryotic cells. The cytoplasm of a eukaryotic cell is a gelatinous liquid containing water, dissolved salts, and other organic molecules. It's also home to several organelles and other structures. For example, the mitochondria, which release energy, ribosomes, which are responsible for protein synthesis, and chloroplasts, which are the site of photosynthesis in plants. Eukaryotes are organisms like us humans which are mostly made up of eukaryotic cells. In fact, every known multicellular organism is eukaryotic, whether it be a plant, an animal, or a fungus. Though some eukaryotes are single-celled, for example amoeba and yeast. Now let's have a look at the nucleus in a bit more detail. The nucleus is a spherical organelle which contains the genetic material of a eukaryotic cell. As you can see in this diagram, it's usually the largest organelle within an animal cell. If we're talking about more than one nucleus, we refer to them as nuclei. When we look at cells under a microscope, we can stain the nuclei so they're easier to identify. If we're looking at onion skin cells like the ones we saw at the beginning of the video, the only reason the nuclei are visible is because they've been stained with iodine, which gives them an orange-brown colour. The nucleus has a complex interior structure. It's surrounded by a double membrane known as the nuclear envelope, which is usually continuous with another organelle called the endoplasmic reticulum. The nuclear envelope is responsible for separating the contents of the nucleus from the rest of the cell, thereby protecting DNA from reactions that may be occurring in the cell cytoplasm. This is called compartmentalization, and it occurs in all organelles which have membranes. The nuclear envelope contains tiny openings called nuclear pores, through which small substances can pass between the nucleus and the cytoplasm, but larger molecules cannot. For example, while DNA molecules are too large to fit through the nuclear pores and so remain within the nucleus, mRNA molecules are smaller, so they can move out through the nuclear pores towards ribosomes in the cytoplasm to carry out protein synthesis. Let's use an analogy to better understand the role of the nuclear envelope. Imagine the nucleus is a school library containing large amounts of information in books, or in the case of the nucleus, genetic material. The doors to the library represent the nuclear pores found within the nuclear envelope. While small objects like books might be taken in and out of the library through the doors, larger objects like the bookcases might be more difficult to move in and out. Within the nucleus is a gel-like substance called the nucleoplasm. The chemical reactions that occur in the nucleoplasm are separated or compartmentalized from the cytoplasm by the nuclear envelope. Notice how the words cytoplasm and nucleoplasm both have the same plasm suffix. This is because the cytoplasm is the liquid portion of the cell and the nucleoplasm is the liquid portion of the nucleus. The nucleoplasm contains DNA in the form of chromatin, as well as many other substances including nucleotides and enzymes which are used in DNA replication. If you're not sure what we mean by the term chromatin, then don't worry because we'll be coming back to it a bit later in the video. The final structure we're going to look at, which is also found in the nucleoplasm, is the nucleolus. The nucleolus is where ribosomes are produced from proteins and RNA. Because the nucleolus is so dense, it's usually easy to spot in a microscope image. Ribosomes consist of a large subunit and a small subunit. These subunits exit the nucleus via nuclear pores and assemble to form a ribosome when protein synthesis needs to be carried out. As we can see in our cell diagram, many ribosomes remain free in the cytoplasm, but some can also attach to the rough endoplasmic reticulum. Molecules of mRNA, which also move through the nuclear pores, will then bind to the ribosomes when specific proteins need to be produced. This demonstrates the important role that ribosomes play in protein synthesis. Now that we've looked at the structure of the nucleus, you might be wondering what makes it so special that almost every cell of a complex organism requires one to function effectively. To answer this question, we need to know about the precious genetic material the nucleus is there to protect, DNA. DNA is a specialized molecule which is usually double-stranded and has a helical shape. Each DNA molecule contains many genes. You may recall that a gene is a section of DNA which often codes for a specific protein essential for the cell's activities. We already mentioned that the DNA of just one human cell spans 2 meters in length. So how does all this DNA fit inside a cell which is so tiny that it cannot be seen by the naked eye? The answer is that it's very well packaged. Eukaryotic DNA is wrapped around specialized proteins called histones, which in turn coil up to build a chromatin fiber. The chromatin fiber further coils up and condenses to form a chromosome, which is located in the nucleus. This process of DNA packaging is a bit like folding up your clothes so that you can fit them all inside your suitcase when you go on holiday. Let's find out a bit more about chromosomes. A chromosome is a single molecule of DNA and its associated proteins which contains genetic material in the form of genes. We often think of chromosomes as having an X shape, but it's important to note that most of the time this is not actually the case. This is what a single unduplicated chromosome looks like. During cell division, DNA duplicates. The duplicated chromosome is still called a chromosome, but now it consists of 2 sister chromatids, which are joined at the centromere. This is why a duplicated chromosome looks like an X when we study it under a microscope. Even though an unduplicated chromosome looks like a chromatid, it would be incorrect to refer to it as such, because it only becomes a chromatid during cell division when it's part of a duplicated chromosome. Chromosomes change their structure and appearance depending on what stage of the cell cycle a particular cell is in. A cell has a life cycle called the cell cycle, which can be divided into a growing phase, I, which is short for interphase, and a division phase, M, where a cell divides either by mitosis or meiosis. Mitosis produces genetically identical daughter cells. It's how an organism grows and how cells are replaced if they become damaged. Meiosis, on the other hand, produces genetically unique sex cells, which are known as gametes. Gametes contain half the genetic material of a parent cell and are needed for sexual reproduction. This is where one gamete fuses with another gamete from the opposite biological sex, and the genetic material of the parents is passed down to the offspring. A typical cell spends most of its life in the interphase stage, making preparations before undergoing cell division. During the first part of interphase, the cell grows. Next, the genetic material on each chromosome of the cell is copied exactly by DNA replication, so the new daughter cells will have exactly the same DNA as the original parent cell. Once DNA replication is complete, the cell grows again and prepares for cell division by mitosis or meiosis. Here is a diagram of some typical cells under a light microscope. To be able to copy the DNA during interphase, it cannot be in the condensed form of a chromosome. So under the light microscope, we would not be able to make out any specific structures. On the other hand, during the mitosis or meiosis cell division phase, the chromosomes condense and are now visible. Remember, the DNA is duplicated in this stage, so the chromosomes have an X shape because they each consist of 2 chromatids. If we zoomed in on one of these chromosomes, it would look like this: 2 sister chromatids held together by a specialized DNA sequence called the centromere. It's the centromere that allows the cell to control the distribution of DNA during cell division so that both daughter cells will receive the same amount of DNA. In most multicellular eukaryotes, the nucleus breaks down before cell division, leaving only a group of specialized fibers called spindle fibers. These spindle fibers attach to the centromeres during cell division to pull an equal number of chromatids to each end of the cell. Once the cell has divided into 2, a new nucleus will form around the chromosomes in each of the new daughter cells. Now we've learned all about the structure of the nucleus and the function of chromatin and chromosomes, let's have a go at a couple of practice questions. The diagram provided is a drawing of a eukaryotic cell. Which structure is indicated by Y? This question presents us with a diagram of a eukaryotic cell. Eukaryotic cells contain a nucleus and other membrane-bound organelles. We can see that our mystery structure Y is located inside a larger circular structure that's surrounded by membranous folds. This larger structure is the nucleus, and the membranous folds make up the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi apparatus. To work out what Y is, let's review the structures of the nucleus. The nucleus houses all of the genetic information required for protein production and other life processes. Genetic information is stored in a long molecule called DNA, which is coiled up into chromatin so that it can fit inside the nucleus. As we can see, the chromatin is represented as long, thin strands, and is therefore not our mystery structure. The nucleus is surrounded by the nuclear envelope, which contains nuclear pores that allow small molecules like mRNA to move in and out of it. The nuclear envelope is directly in contact with the rough endoplasmic reticulum, and is therefore also not structure Y. The final component of the nucleus is a large, dense structure called the nucleolus. The nucleolus is the site of ribosome synthesis. You may recall that ribosomes are the organelles that are responsible for synthesizing proteins. We have therefore solved the mystery and deduced that structure Y is the nucleolus. Let's try a second practice question together. How do substances pass between the inside of the nucleus and the surrounding cell cytoplasm? A. Through nuclear pores. B. Through gaps in the nucleus wall. C. Through the nucleus stomata. Or D. Via nuclear transport tissues. The nucleus is a membrane-bound organelle which contains the majority of a eukaryotic cell's genetic material in the form of DNA. Its function is to protect DNA from the reactions that occur in the cytoplasm, and it does this using its membrane, which is called the nuclear envelope. However, some material must pass between the nucleus and the cytoplasm in order to give the cell essential instructions. So how does this happen? The nuclear envelope contains small openings called nuclear pores, which some molecules can pass through. Chromosomes are unable to pass through the nuclear pores because they're too large. mRNA molecules, on the other hand, are much smaller, and therefore they can pass through easily. Thanks to nuclear pores, mRNA can pass from the nucleus into the cytoplasm, where it can meet a ribosome, and together they can carry out protein synthesis. We have therefore determined that the correct answer is A. Substances pass between the inside of the nucleus and the surrounding cell cytoplasm through nuclear pores. Let's recap some of the key points about the nucleus that we've covered in this video. The nucleus is a membrane-bound organelle which contains an organism's DNA. Eukaryotic cells contain a nucleus and other membrane-bound organelles, whereas prokaryotic cells do not. The nucleus is surrounded by a nuclear envelope containing nuclear pores. The nucleoplasm is the liquid portion of the nucleus, which contains the nucleolus and DNA in the form of chromatin. Cells have a life cycle called the cell cycle. And finally, during cell division, chromosomes are duplicated and consequently have an X-shaped structure consisting of two sister chromatids.